Good afternoon, everyone. So I'd be delighted to tell you in a quick overview what goes on in my lab, my group, what has been going on for the past 5 years, including this year at SDSU, my first year here. So what I study is the biosynthesis of the modified nucleosides of a specific molecule that I'm interested in, which is an RNA molecule, which is the central molecule of the central dogma that Stanley talked about. So that molecule looks like this. Funky L shape, and we tend to color it. And there's a reason for that, because we like to have different colors for different areas of it. In real life, it looks like this, and it exists really in all life forms. There would be no life without it. And when we want to understand it and draw it and communicate about it, we expand it into this shape, which is the clover leaf shape. And each one of these arms have loops around them, are there to serve a certain function. And people have focused for a long time on exactly how this molecule folds and how it is made and how it is modified, right? So when it folds in 3D, it looks like this L, where you can see the different parts in color have gotten together to correspond to basically the same colors in the figure, in the three-dimensional structure. So what you see here, these little these little protrusions here in this model or this particular type of rendering of the molecule is these are what we call nucleosides. These are the building blocks, the units that this molecule is made of. And there's usually about 73 or 72 of them. So it starts with this end, which is we call the CCA end. And that stands for it's the three nucleotides that make that tip of it, which is the most important tip for its function. That is the end that gets charged with an amino acid that is then carried over to what we call the translational machinery. What is that? The translational machinery is this little two bubbles, which is hyper-simplified in this view, that really amounts to how our genes are translated. All DNA and all life forms are translated at the end by the action of this machine that's called the ribosome on a type of RNA called messenger RNA, which is really what acts as a transcript of the DNA of life, and is carrying the information or the genetic code that encodes particular proteins, which make us all at the end, right, which is what we all are made of. So that mRNA gets grabbed by this machine, and then that same machine, the ribosome, holds on to another molecule, which is our tRNA, the transfer RNA. And it uses that to decode the messenger, to decode the message, the nucleotide triplets in the mRNA to their amino acid correspondence. And then that machine ends up synthesizing a polypeptide chain from these sequential amino acids. So it binds one tRNA, another, and a third, 3 at a time, to connect the amino acids and enable the formation of a polypeptide chain coming off. What we are interested in is this molecule, is the tRNA itself. How is it made? We always thought that it's a string of nucleic acid or nucleotides, and those nucleotides are 4 types. But not really. We learned a lot about it in the past, perhaps 50 years. So RNA in this depiction is made in the nucleus of a eukaryotic cell, an animal cell, for example. And it's here made in the nucleus, and it has to make it through the nuclear membrane or wall to come out and participate in the translational machinery. But before it does that, it gets heavily processed. First, it has a bunch of extra pieces that are unnecessary for its function, so far we think. We call introns. They get removed. And then it gets edited, meaning some of its nucleotides get swapped with something else. And then it gets modified, and it gets trimmed at this 3' and 5' ends, the tips of it. And then that tip at the 3' end gets a CCA added to it, that triplet that is necessary for its charging or loading with amino acid. All of that goes on the nucleus and it gets transported through the nuclear pore complex, where it gets carried over by this huge protein complex called the multisynthetase complex, where there's a conglomerate of enzymes that charge it with their corresponding amino acids. So it grabs a bunch of these tRNAs, And then the tRNA is delivered to the ribosome where it gets used for translation. It gets recycled, into this fashion with a certain lifetime. What we're interested in is how this molecule is made. And of course, we know that it's modified, and that has been the biggest dilemma. What are these modifications? We find that when you take an average tRNA from a cell, it doesn't have just the canonical nucleotides. It has nucleotides that are kind of looking funky. They have these additional chemical groups that are added. There could be small things like methylene, for example, or a sulfur atom. Or it could be large and complex chemical moieties, that like a whole amino acid in this case, for example. And these modifications occur at what we call the base, right, or the nucleobase of the tRNA, of the nucleoside. And these tend to cluster in areas like here at the bottom, where it is called the anticodon. This is the area where that is actually interacting with the messenger and decodes the messenger. So it's highly important for function. This is another area which we call the elbow, where also these modifications tend to cluster. And you can see the small ones tend to cluster here, the large modifications tend to cluster here, and there's also functional reasons for that. These here tend to be necessary for the thermal stability of tRNA, especially in organisms that live at very high temperatures, such as the volcanic vents at the bottom of the Pacific Ocean, or over there near Italy. So this is tRNAs in these organisms highly modified on the elbow. So we're interested in how are they made, but before that, I want to give you an overview of what they're for. I mean, there are numerous functions that have been found, but what the most relevant ones that most people are interested in is their role in HIV replication. Several of these modifications have been implicated directly in the ability of HIV to replicate inside of our immune cells. And there's a myriad of metabolic functions that these modifications are necessary for, in addition to things like, as I said, adaptation to high temperature. And the most recent findings in the past, perhaps 7 years, has been the role of these modifications in the development of neurodegenerative disease. And you can see, it's not here. Oh, the human mitochondrial energy cycle. There's one modification, is the TM5U, has been implicated in that function. And human mitochondria in the brain are necessary or have been, of course, the primary cause or malfunction there has been the primary cause of neurodegenerative disease. So how are they made? We were interested in that. But we focused particularly on two modifications. Quiosine, which looks like this. It's a guanine base that had lost that nitrogen here and has a huge moiety added to it. And another one we call T6A. It has this long name. And it's basically an adenosine, which is the basic nucleobase that had been modified with a carbamoyl and a threonine moiety, which is an amino acid. And the question was, how are these modifications made? It turned out that there's a number of enzymes. So we discovered a number of enzymes involved in pathways that could involve 10 to 12 enzymes at a time. So I'll give you examples of that. But the question is, what is our approach to this kind of discovery? Basically, the question is, what are the genes encoding the enzymes or the proteins that are involved in the biosynthesis of these modifications? And the approach is really tripartite. We identify gene candidates using a method called comparative genomics or structural bioinformatics. It's this computer-based type search, very detective work type. And then we make hypotheses from that we can then experimentally verify or check, prove or disprove by doing genetic manipulation, knockout experiments of these genes. And then we can then clone these genes if we actually get a hint, and we end up doing some enzymology work in test tubes, basically, to verify function that we predicted. And we go back and forth in all directions, so it's an iterative process. And the discoveries we end up making from this kind of work, not only do we identify the biosynthesis pathways for these modifications, but we discover new chemistry. And we discover new drug targets, specifically and against infectious diseases. And we uncover new ways that pathogens resist the immune response. So this is an unexpected path that my lab has taken just this year at SDSU. And this is, I'm going to just give you a few examples of these 3 different types of discoveries. For example, this is a pathway that we focused on for the past 5 years. It's a pathway for the biosynthesis of the modification quiosine, which is this one here. And in bacteria and archaea, which is the third phylum of life, they all get biosynthesized using a common pathway. And then it splits. In the bacteria, it goes one way, in archaea it goes another. But what I want you to focus on is these few enzymes that we discovered and we studied heavily. The first is the first enzyme in the entire biosynthesis pathway. And you can see there's 1, 2, 3, 4, 5. There's 5 at least, or 6 enzymes in the bacterial pathway. And there's maybe also same number in the archaeal pathway. So we discovered the GTP cyclohydrolase 1B as the enzyme responsible for catalyzing the first step in the pathway. And another enzyme, which is QF. And the third is the QF-like, and ARC-S, and GAC-QC. I will focus just on a few of these examples. Okay, forgot about that. So QF turned out to be the only nitrile reductase known in biology. It looks like this. We call it a tunnel fold, and it has 10 subunits. These ribbons you see are basically just a rendering of the alpha carbon chain of this molecule. Turned out that this molecule is regulated by disulfide formation because it's highly redox sensitive. It's a redox enzyme itself. And we did a lot of work this year to prove that this is, in fact, a regulatory mechanism of the function of the enzyme. Another is the QF-like, which is actually an archaeal enzyme, which is also a tunnel fold that had evolved from a pre-QF ancestor. It's an aminotransferase, and it's the only type that interacts with tRNA. So that's in itself is novel chemistry, unexpected. The third is the drug target, which is a GTP cyclohydrolase. It's also a tunnel fold, so we have 3 tunnel fold enzymes from our lab. And that is now a drug target. Why? Because it's distinct from a human homologue, and it makes, and we could actually investigate the details of its active site by probing it with inhibitors. And now we are doing a structure-based design method or approach to designing small molecules that can block its active site and block it from working. So this is a drug target. Why? Because it's responsible for folate biosynthesis in a certain subset of pathogens. More drug targets we could discover is from the T6A pathway, which actually has 4 enzymes we discovered that are responsible for it, the TSA, C, B, D, E. And we could build a model of the complex formed by these proteins. And only recently, my postdoc, Amit Luthra, who's sitting here, managed to get a SAX, or small angle X-ray crystallography structure of it, showing pretty much kind of that my model was correct. And I will stop here, and I'll take your questions.
Questions for Manal?
So Manal, how close are you to taking any of these potential inhibitors and turning them into something that might be an effective small molecule therapy?
I would say we're 5 years away. Why? Because we have so far in the first tier or level of compounds which have been predicted to bind in silico, meaning at the computer level, and we've screened about a third of them. And from just that third, we landed 7 compounds that inhibit in vitro. And although they are not at the right intensity or efficacy, but I think just the rate of success just from that first stage is pretty promising. So I think within a few years, we could have a compound or 2 that would bind in sub-micromolar in KD or binding affinity. Go ahead.
Hi, very interesting. I have a quick question about the ribosomes or the modifications take place within the nucleus that you're speaking of, where the
Yes,
small
they do.
molecule inhibitors would be inside the nucleus. So you would need to find a small molecule drug that penetrates the nuclear
not
membrane?
in this case. It's just so happened that this first enzyme, which is the enzyme for the modification, making the modification creatine, is also the first enzyme in the folate pathway. I didn't show that, and there's even a third pathway in humans that this enzyme is also responsible for. It's a type of reaction called GTP cyclohydrolase reaction, and in bacteria there's no nucleus, so we don't have to worry about it. But it is a bacterial enzyme, and we're interested in inhibiting it.
Anyone else?
That's very nice, Manal. I have a quick question, just a general question for you. So I guess of all the different types of RNA molecules, is there a preference of which of the three main types are actually have these different kinds of modifications? And is there preference of different types of modifications within tRNA, RNA,
Yes.
mRNA?
Yes, there are these modifications, not all of them, but modifications exist in all RNAs, including messenger. This was actually discovered 2 years ago, not even, less than 2 years ago. Exist even in small nuclear RNAs. And in fact, my collaborator found that they exist even in DNA, including phage DNA, which is yet another project we added to the lab. But yes, to answer your question, tRNA remains the prime site or molecule that harbors the maximum number and diversity of these modifications. Mm-hmm.